Thank you, Dr. Olson, for that very nice introduction, and welcome to San Francisco, everyone. It's a pleasure for me to join you here this evening to kick off your meeting. And as you just heard, I've spent most of my career thinking about product development and about moving interesting science into clinical trials. And so I'm going to attempt through a number of stories and maybe a few pearls of wisdom, at least I hope they're pearls of wisdom, to convey to you how I think about moving cutting-edge research into the clinic. And particularly, I hope that if you take nothing more away from this talk than this, it is an enormous thrill to think that you can take advantage of great science and great intellect and make a difference for patients. And while it's challenging to do so, if done well, it is one of the best things that any professional can do. And so I'm going to have 4 simple areas that I'll address for you this evening. I don't like to only tell positive stories. They're always my favorite stories. And as you go through the challenges you have with your programs, you'll find one of the lessons that I've always carried, which is failure is an orphan. But success has many mothers and fathers. So I will tell you a negative outcome story, and you won't find anyone who was participating in this, and then a positive outcome story which has hundreds, if not thousands, of champions. Some lessons that I've learned from trying to make products available for patients. In my new job, I've thought a lot about the interaction between companies, the venture world, and academia, and I'll give you a few thoughts I have, but I would love in the question and answer session to hear your thoughts. What's academia's role? How does academia fit into moving innovations into the clinic? And end with a little bit of thinking of what's possible in the future and maybe some directions that we might want to take to make what's possible in the future come sooner and better. Let me start with a few perspectives that I have. One of the things that I noticed when I first began speaking about moving discoveries into clinical trials in academia is that I had a very specific perspective about this that was a bit different than some of my colleagues. I define the moving a discovery into clinical trials as a step. It's a very important step. It's worth celebrating. It's wonderful, particularly if you've been working hard on science for a long time. So filing an investigational new drug application with the Food and Drug Administration or its equivalent outside the United States is a thrill. And having the first patient treated with your potential new therapy is great. But that's not the goal. It's an important step, but not the goal. For me, the goal is regulatory approval, if it's in the United States, Food and Drug Administration approval, and product commercialization. And the number one challenge in doing that, when you're focused on the discovery, is often efficacy. So we always think about decreasing suffering, curing, improving outcomes. But in fact, the number one challenge is safety. It is doing all those things, improving outcomes, benefiting patients, while making sure that we don't harm patients or that the net benefit is not there. And during clinical trials, we always increase the number of patients incrementally. But in fact, more and more, particularly with chronic conditions, proving long-term safety has become the challenge for any new therapeutic advance. And this is particularly true, and I think it's good news that more therapies are available for patients. But in those cases where existing therapies are available, it's our challenge in the world of translation to ask ourselves, am I very, very confident that my therapeutic advance is better than the alternative for the patients? So the reason I bring these perspectives is really a couple of things. One is I don't think that it is as important to treat one patient at a time, although each patient contributes an enormous amount by agreeing to be in a clinical trial. But we should reach the greatest number of patients and have the greatest good for society. And so when you achieve regulatory approval and commercialization, that allows many, many patients, no matter where they are, to potentially have access and have the greatest good. Whereas in a trial, there could be promise and it could be helpful, but the number of patients by virtue of a trial is always limited. And I'll say, and I know that you know this, and I knew this when I first started in product development, but it is essential to remind yourself constantly of this. Innovation has its good, and we're always so hopeful and optimistic in general when we're approaching clinical trials, but all of our innovations can be harmful. And not just harmful in the obvious ways that we measure in clinical trials, but long-term can have effects that are harmful. And this is particularly true when you're doing something that's never been done before. And as thrilling as it is to do something that's never been done before, keep your eyes open to things that you never anticipated because it's never been done before. So I want to tell two stories about two therapeutics that I worked on during my time at Genentech. And both of these therapeutics were molecules that I had interacted with the scientists in research, in basic research. And during my tenure at Genentech, we filed for the first clinical trials, went through the clinical trials, in one case to an approval, and in one case to a phase 3 failure. So the first story I want to talk about is nerve growth factor, a protein therapeutic, a growth factor, which we were attempting to develop as a potential therapy for diabetic neuropathy. And this molecule had success in phase 2 and failure in phase 3, and I'll talk more about that. And the second molecule is a monoclonal antibody, bevacizumab or Avastin, an anti-angiogenic and anti-vascular endothelial growth factor for cancer. That the concept was published in '71, and then we filed the IND to start the clinical trials in 1997, and I'll talk more about that. So nerve growth factor is it's actually not an antibody, I'm sorry, that's a typo. It's a protein that is known for a long, long time to have effects on nerves. There's also an antibody, anti-nerve growth factor, that remains in development that blocks nerve growth factor. So NGF was originally cloned at Genentech in 1983. And in 1996, based on positive phase 2 results, the protein therapeutic was advanced to phase 3 trials. And in 2000, 2 randomized, blinded clinical trials compared nerve growth factor to placebo in about 1,000 patients, and both these trials were negative. So enormous amounts of time, energy, and patient time were spent. The technology, both the nerve growth factor and the antibody to nerve growth factor, were then licensed to Rinat in 2004. Not a good story. So why do I talk about this story? I think we learn as much from the failures in the clinic as we do from the successes. So let's start with phase 1, and let me talk for a minute about the decision to go into phase 1. So nerve growth factor was known to have an impact on growing nerves, that's the name. In preclinical models, which weren't ideal, there was a suggestion that there could be some nerve regeneration and therefore a positive outcome in diabetic neuropathy. Diabetic neuropathy had no therapies. There were no approved therapies. The treatment was symptomatic, and so it was a very large unmet medical need. When we did phase 1, the phase 1 initial dose caused severe myalgias, and it was the first phase 1 study I had done, particularly given my background in cancer, where we started low and always made our way up, where the first thing we did was lower the dose. So we started above what turned out to be the maximum tolerated dose. So the first lesson was to always remember that dose ranging done in preclinical models or in animal models is a suggestion, is at best your best attempt to look at no effect doses or maximum tolerated doses, and be open to adjusting doses up and down. The other lesson is something early on, the myalgia that we saw suggested that an antibody to nerve growth factor, which now is in clinical trials, might actually have a positive impact on pain. Phase 2 looked positive. Phase 2 trials were done uncontrolled, no placebo, open label trials in patients with diabetic neuropathy. And it looked incredibly promising. The patients reported that their pain improved when short-term treatment with nerve growth factor was administered. They did have some very mild myalgias at the time of administration, but the lesson seems to be relearned every time I read about new molecules. The value of controls and the value of blinding. Very hard to do with therapeutics that have adverse events. But the placebo effect is especially key when you're looking at a pain endpoint and in chronic diseases, and I would say particularly true in many of the chronic diseases we try and treat today, which have waxing and waning courses. Whenever I see benefit in those kinds of diseases, I ask, and you should all ask, is this the waning? Is this my therapeutic, or have I caught those patients on a waning, or have I seen a placebo effect? So we saw what in retrospect was clearly a placebo effect, because in phase 3, 2,000 patient trials, absolutely negative, no benefit to treating patients with nerve growth factor. The other thing that I think is the lesson I took away from phase 3, because this was an unmet need, because there was no therapeutic approved, we were very, very aggressive in wanting to pursue Food and Drug Administration approval. So we did these 2 phase 3 trials, 2 trials because that was what was required from the Food and Drug Administration for approval. But think of the number of patients who were put on an inactive therapy. Happily, there weren't serious adverse events in the trial, so we didn't harm these patients, but patients signed up to be on a study for a long term who could have tried different therapy and didn't need to be in this trial. So I always think in any trial you do, you want to avoid exposing hundreds of patients to inactive therapy. And had I to do it over again, I would have probably pushed for a blinded placebo-controlled phase 2. It's hard to go slow in phase 3, and it's hard to do interim analyses in chronic diseases and in long-term studies. But I think had we had a placebo, we would have surely seen that in the end, the placebo effect and the nerve growth factor effect were in fact identical. I will say, because you're all working on a remarkable potential therapeutic advance, that at the time that nerve growth factor was in the clinic, Genentech was also working on VEGF, vascular endothelial growth factor, and IGF-1. And in my mind, I still to this day have not seen those protein-based growth factor therapeutics become important medicines. And so I think that this technical approach to treating patients has its challenges. Very short half-life, and a very tough ability to grow or regrow either endothelial cells, blood vessels, or nerves in a way that's meaningful and helpful to patients. So there's a lot to be learned from the experience that Genentech had and that of others in attempting to develop growth factors for chronic conditions. We attempted VEGF for coronary artery disease and also negative outcome in phase 2. That's where you want negative outcomes to happen, not in phase 3. So that's the negative story. A lot to learn, relatively short development program, I would say relatively efficient, but not a positive outcome for patients. Now let's turn to one that was a positive outcome, a lot better story. So anti-VEGF bevacizumab, or Avastin, was the first FDA-approved anti-angiogenic cancer therapy. And I think this is a story that is an amazing story for a number of reasons. So anti-VEGF is a humanized monoclonal antibody. And the first thing I want to say about anti-VEGF that I think is something to think about as you're approaching new therapies for patients is it is a very precise inhibitor of VEGF. There are many small molecule tyrosine kinase inhibitors either approved or now in clinical trials. as anti-cancer agents and anti-angiogenics. What I, to this day, liked best about anti-VEGF is that it has a precise target, and it has side effects, important side effects that we learn in the clinical trials, but those side effects are on target. When you're in a very new area, blocking VEGF to attempt to help patients with cancer, the opportunity to have a precise weapon is a huge asset, because everything that happens, good or bad, is related to VEGF inhibition. When you have a multi-kinase inhibitor, that is much more challenging when you're asking, where does the efficacy come from, and where does the toxicity hail from? Although you can have multiple benefits with multi-targets, much more challenging when you're in a brand new area. So I remember going to cancer meetings in the 80s and hearing Judah Folkman, who unfortunately passed away last year. He published in 1971 and subsequently virtually every year gave a lecture at the cancer meetings. And at some point we all said, there he goes again. And his hypothesis was that a tumor couldn't grow larger than a BB, couldn't be more than 2 millimeters, and it couldn't spread in the absence of new blood vessel formation. So the hypothesis was very clearly that you could starve the tumor, prevent metastatic disease, prevent progression by interfering with new blood vessel formation. But it wasn't until 1989 that Napo Ferrara at Genentech, and in addition, Hal Dvorak identified vascular permeability factor, which ended up being identical to VEGF. Vascular endothelial growth factor was identified as the most potent growth factor for growing new blood vessels. This gave us a potential weapon to use in this hypothesis, as described in 1971, and to form a monoclonal antibody, anti-VEGF, that we put into clinical trials in 1997. So one of the things that is easy to forget now, because Avastin anti-VEGF has been around for much of my career. This has been something I've been thinking and working on. But in 1997, I remember talking to Gwen Fife, who was my colleague and the head of clinical oncology at the time. We had a discussion about the first patient that was going to be treated with anti-VEGF. And the discussion was one that, even though we had been through all the preclinical, even though we had done all the tox studies, and it looked like our hypothesis, which was adults don't use VEGF to sustain their blood vessels, adults in the absence of pregnancy or significant wound healing could be treated with this very potent blocker of VEGF, and we could get away with it. We didn't know for sure. And we were terribly worried about every one of the patients who were treated in phase 1. Well, it turned out that every one of the patients treated in phase 1 survived. They didn't have some acute problem as a result of anti-VEGF blockage, but as we got into higher doses, we reliably caused hypertension and proteinuria. So the maximum tolerated dose of anti-VEGF we saw in breast cancer patients treated at the highest doses we used in phase 1, and it ended up being nephrotic syndrome. Patients definitely had impact on their capillary beds, on their kidneys, on their blood pressure, and you could monitor that acutely in the clinic. So the clinical trials of anti-VEGF in phase 1 showed that the medication was relatively well tolerated, and we did something that was unique in oncology at the time. We did randomized controlled phase 2 studies. They were relatively small, so not definitive, but unlike the experience I told you about with nerve growth factor, we had controlled studies in phase 2. Very, very important for this incredibly novel way of treating cancer. And we could use controlled studies because everyone got gold standard therapy, and the treated patients got gold standard therapy plus anti-VEGF, bevacizumab. So 1997, we filed the IND. We started the clinical trials. And in 2004, the first approval for Avastin in colon cancer happened. In 2006, an approval in lung cancer, a conditional approval in breast cancer in 2008, kidney cancer and brain cancer in 2009, and this week, this last couple of weeks, 2 announcements were made. One, that these gastric cancer trials were negative, and the second, that the ovarian cancer trials were positive. So this may be, in the end, one of the most important advances in cancer in terms of this blockade of angiogenesis, first described in 1971. Now, over 500,000 patients have been treated in the first 5 years since this medicine became approved by the Food and Drug Administration in 2004. So a great story, a positive story in terms of clinical benefit, and the colon cancer trial and lung cancer trials had survival benefits seen in both of them. I have to say, the story's not over yet. Unfortunately, the first trials done with anti-angiogenesis in the adjuvant setting in colon cancer were negative. And so for most of us, we hope that we can put cancer therapies in the earliest form of treatment and have a major clinical benefit, and that hasn't yet been shown with Avastin or anti-VEGF. The other thing that keeps me from declaring victory with these trials is think about that timeline. Description in 1971, we have the first clinical trial in 1997, and here we are in 2010, and we're still trying to see if we can make a major impact in cancer with this form of therapy. So one of the areas of product development that I think we all need to think about is the pace and the efficiency, and I'm going to come back to that because I think even the great stories should be better, faster and better. So how do we improve our ability to translate discoveries into benefits? How do we see the kinds of outcomes we all dream of seeing in our lifetimes? Well, I have 3 things that I would put forward as areas that I would love for this group to think about, because you're all innovators. You're doing something people have dreamt of doing and have never done before. And as you enter clinical trials, these are some things I think are worth thinking about. Technical aspects that I think we could do much, much better. And the first is reverse translation. It's kind of a jargony thing to say reverse translation, so forgive me if you don't like the term. But it allows me to describe something I believe we don't do often enough. I like to tell people that my favorite animal walks upright on 2 legs. I'm not a basic scientist. I'm a clinician by trade, by heart. And all my energy when I hear about biology or science goes to thinking, how will this help patients? I think a lot more of our energy ought to go the other way. Every time a patient has an experience in a clinical trial, we should learn from that. I just told you more than 500,000 people have been treated with anti-VEGF for cancer. Well, as healthcare reform happens, as electronic medical records come about, we should learn from every patient treated with a novel therapy. And I just told you something about VEGF inhibition, about hypertension, proteinuria, nephrotic syndrome. I didn't tell you, but 2 of the most important side effects of long-term therapy with anti-VEGF, particularly in those over 65, are stroke and MI. So think of all the time, energy, money spent on knockout mice. Well, we've got over 500,000 human beings treated with a very, very precise inhibitor of VEGF, a monoclonal antibody. Think about the biology that can be learned from those patients. Every time we do a clinical trial and we intervene or make some new intervention for patients, both positive and negative outcomes occur, and we don't measure the half of it. So I think every time we have a new therapeutic, not only for safety, but for efficacy, and more importantly, for understanding biology, we ought to be going back from the bedside, back to the bench, and learning more about the biology that we're intervening on in the clinic. This is something that doesn't get much investment, doesn't have a lot of time at companies, but I think we could do much better and much more with this. And not to mention mechanisms of resistance to therapies once we're in the clinic. So improved translation, and this is Joe DeRisi, one of the superstar scientists at UCSF, who's an expert on all things array at bioinformatics and using genomics to really improve how we think about patient therapies and about science. So it's very popular to talk about personalized medicine. It's kind of a vogue thing. And like many things, I think the term personalized medicine has been overused or misused. But here's what I mean by improved translation. There are patient populations that are big and broad in chronic diseases like diabetes, like Parkinson's disease, like many cancers, where if you think about bringing a new therapeutic into those patient populations, it's daunting. There are active approved therapies. The number of patients it would take to prove that your therapeutic advance is better than the gold standard is enormously challenging. The ability for us to subset patients with diseases into different categories based on genomics, based on arrays, is an enormously positive tool for product development, for drug development. We can do two things. We can make sure we do get the right therapeutic to the right patient at the right time, but even more, we can be much more efficient with our clinical trials. We can target the patients for whom today's therapy doesn't add enough value, and we can be very precise at saying these are the very patients who don't have something else. There is no gold standard. So the ability to subset patients who have the biggest unmet need, whose disease advances the most rapidly, and who might be just right for the therapeutic advance we have, is an enormous asset on your timelines when you do go into the clinic. Another area that I think we can have more effective clinical trials is our outcome measures. And this is just an example of an X-ray. And the difference in looking at plain films or MRIs as compared to some of the new MRI and PET scanning approaches that are available now. What I want in a clinical trial is I want to know that the patient has too much of something is dialed up, I intervene and I dial it down. And I dial it down at the site I care about. And the ability to read out our intervention on making something go up and down quickly has enormous potential benefit for patients, instead of keeping patients on 1 year, 2 year, 5 years on a trial. And I think imaging, as well as some of the proteomics and blood-based markers, show the greatest promise for rapid readouts. There are patients who aren't going to benefit from our therapies. We should be able to find that out before looking at things like survival, long-term outcomes, where it's too long for patients to wait and for us to wait for those answers. So the use of imaging, and particularly the use of metabolics in imaging and molecular imaging, I think will greatly advance our clinical trial timelines. So where does academia come in? There are people who say, you shouldn't be doing product development in academia, that's what companies do. And I do think that's what companies do when it comes to commercialization. That's the job of companies. But universities stimulate curiosity and create knowledge. And companies develop and commercialize products. That we each have our role. What I think is so special about being in academia when it comes to translational research is we can pursue technical excellence. We can ask questions because that knowledge can apply broadly to patients regardless of commercializing a molecule or a compound. And I think one of the areas that was the most interesting with AntiVEGF, most recently we were developing AntiVEGF for glioblastoma, for brain cancer. And a question came up that was extremely important. Are the effects of AntiVEGF on brain cancers anti-tumor or anti-edema? Now you might say, and I might say, that's kind of a theoretical question. Does the patient feel better? Do they live longer? Do they have better outcomes? Is there clinical benefit? But that's an essential question. Does this molecule that is anti-angiogenic, not affecting the DNA, does it actually kill the tumor cells? Well, that's a great scholarly question that could help advance the field of anti-angiogenesis for brain cancer, but also has broad biologic impact on how we think about this really difficult to treat neoplasia. So I think those are the kinds of things we can do in academia. So I want to tell you briefly about 3 translational programs emerging at UCSF that I think are very exciting, and the kinds of things that are happening broadly in academia, but I know our programs best. And let me just describe for you quickly these 3 programs. So the Clinical and Translational Sciences Institute, or CTSI, under Clay Johnson, promotes rapid application of discoveries to prevention, treatment, and cure of human disease in the clinic and the community. What I like best about this program is it has training and infrastructure. There are online tools, guidelines for ethical and regulatory issues, funding assistance, consultative services, and clinical research centers across the UC system. This is a program that I actually trained in before it was called a CTSI. There was a summer course, and I designed my first clinical trial in that summer course. These are the kind of practical things that make a huge impact when people know how to access that kind of training. And it's fantastic for students and for trainees, but also for career development. So one thing that the CTSI does is they have KL2 Career Development Program that's a 4 to 5 year program for a select number of scholars at a faculty level with salary support. It includes a Master of Sciences degree in clinical research, and it emphasizes both T1 and T2. So we've had people go through this who are leading thought leaders now and faculty members. So these are the kinds of programs that early on in a career can change the outcome for someone in terms of how they think about their ability to operate in a translational mode. I often tell people when I went through my training, I spent a couple of years in Uganda working at the Uganda Cancer Institute as a UCSF faculty member, a couple of years in private practice as what my parents call a real oncologist, 2 years at Bristol Myers Squibb, and then I come to Genentech. Well, in my own way, I had translational training, but we could be a little more purposeful about that. And that's what CTSI ensures, is that you get systematic, great training in everything it takes to be a great clinical investigator. We also have QB3, and this is actually a partnership between UCSF and it's UC Santa Cruz and UC Berkeley as well. And this is a fantastic program that is a partnership between the state of California, private industry, venture capital, and it's bringing our discoveries to society. So it emphasizes industry partnerships. We've got 16 garages over in Mission Bay where small startups can have a lab bench. We have knowledge brokers, an incubator network, and clinical associates. And this program, again, allows insight for people without this kind of experience in what it takes to bring something out into industry. And one example that I think is a fantastic example is Chagas disease, the most serious parasitic disease in Latin America. This is a neglected disease and hasn't been pursued by pharma because it's a small number of patients. But QB3 brought together experts in drug development, regulatory affairs, industry partners, and now there's a compound, K777, that could actually cure Chagas disease, developed entirely within the university, but with help from industry, in a collaboration that I think is, in my experience, unprecedented to bring forward something that could have a huge benefit. The last one I want to describe is a new inter-school department at UCSF, and this is the Department of Bioengineering and Therapeutic Sciences, and this is a fantastic collaboration that includes pharmacology, engineering, drug development sciences, computational and systems biology, educating PharmDs, MDs, PhD grad students in bioengineering and therapeutic sciences. I often think that we could learn a lot from engineers in predictability. It's a very innovative program, and one of the aspects I want to point out for all of you that I think is one of its greatest assets is this course, the American Course on Drug Development and Regulatory Sciences, which allows people to learn how to do something that isn't often even described, which is training professionals to work on all sides of the product development pipeline and gain in-depth knowledge of how you do this. What is an IND, an NDA, a BLA, phase 1, 2, and 3? What does all this mean? What are the rules? And how do I navigate all those rules? And the ultimate benefit of this course is better and safer medical products. So these are just 3 examples of what we're doing at UCSF to think about that translation and how much training and expertise and technical advances need to be had. So knowing that I was going to come and give you a talk, I looked over the some of the material that you all have in terms of your objectives, and I really love this, to support and advance stem cell research and regenerative medicine under the highest ethical and medical standards for discovery and development of cures, therapies, diagnostics, and research technologies to relieve human suffering from chronic disease and injury. What a great set of objectives. So I thought I'd give you my thoughts on the objectives, just given the experiences I've had. I would push you to ask, does the approach you're taking offer the highest likelihood and fastest route to relieving human suffering? It's such a great objective, and I've always found if I push myself and have as my personal rally cry, patients are waiting, it pushes me to do good things. I have a sense of urgency for the right reason, and I think relieving human suffering is an absolutely worthy goal. But also, and I kind of get back to this reverse translation, ensure you learn something from each and every trial carried out. And when you do a trial, the answer might be yes, or it might be no. But the real tragedy, and all of us who interact with patients in clinical trials, the answer should really not be maybe. And if it is maybe, I always ask myself two questions. Am I being intellectually honest? Is it really a no? And I'm hoping it's a maybe. And did I do a lousy trial? Did I subject patients to a clinical trial that didn't get a clear answer? Because that's our job when we ask patients to volunteer for a trial. Every patient who enters any of your trials, both short and long-term, provides enormous amounts of information that can benefit other patients. And the final thing I'll say about my thoughts, and I push myself on this, and I think we all need to push ourselves, it's an era of healthcare reform. What can we do in our particular project and our focus to make our approach as simple and as inexpensive as possible? Patients aren't going to benefit from our therapeutic advances if we keep increasing the complexity and increasing the price. So therapy should be scalable, and in the end, if patients don't have access to it, it doesn't count. So it needs to be scalable and accessible. And I most like all of your objectives because I think what's really exciting about what you're doing, turning biomedical discoveries into products that benefit patients, it's not just gee whiz science, which it certainly is, but it's the power of public funding, the partnerships that I know you're all setting up, and using your own intellect and wisdom and passion to make a real difference for patients and their families. So I'll end by just saluting all of you and CIRM and the work that you're doing, and thank you for inviting me. I did leave time for questions or comments, I think around 10 minutes or so. And there's 2 microphones on either side if anyone has any questions. I hope you have a few hard questions, or I'll really be disappointed. There's someone over here. Can you say your name and where you're from so I know who's asking?
Sure. Ed Minucci from UC Irvine. So I was really interested in the reverse translation idea. And I remember clinical colleagues of mine a number of years ago talking about one of the things that was the least satisfying to them about clinical trials was the opportunity to do, to get scientific data back from the patient, but the inability to do that because of the restrictions on the kind of data you might be able to get back from the patient. And so, like you said, you have over 500,000 patients treated with anti-VEGF therapy, and what sorts of information can you get back? But I guess my big question is, has there been a change in thinking about, loosening up what kind of information we can get back from the patient so somebody can actually do real science on the humans, on people getting treated?
So I would give 2 answers to your question. The first one is that the logistics and the limitations still exist. The best example is in oncology, we often would like to have pre and post biopsies, right? I mean, if you make an intervention, you would love to know what the tumor and all of its various growth factors and markers look like before your intervention and one after. That can be an enormous impediment to enrollment. Patients need to consent separately for a biopsy. So the practical considerations haven't changed. what I think remains true, and what I've seen done in a very positive way, is the ability to collect data to form hypotheses is absolutely possible. So the FDA is asking many companies for what are called REMS, these post-approval close monitoring for safety. So patient registries are being used for virtually every new approved therapy. Well, the amount of information in those patient registries is huge. And what we started to do when I was at Genentech is you can embed a sub-study in a patient registry. And so post-approval, you can actually do studies where you consent patients who are in the community and receiving your therapy to do additional science. I think it's much more likely to do that post-approval than in phase 3. But the other thing that I think we should all push ourselves to ask is, patients actually undergo a number of procedures as part of their normal clinical care, and including a lot of data collection. So as we have electronic medical records, I think with appropriate attention to HIPAA rules and all the confidentiality, it is possible to continue to collect data. I know I didn't talk about it today, but Rituxan, which is one of the most amazing antibodies that I ever worked on. So Rituxan is an anti-CD20 monoclonal antibody approved for non-Hodgkin's lymphoma and chronic lymphocytic leukemia. And over time, we did a number of things, both in the clinical trials, but also in post-marketing non-Hodgkin's lymphoma registries, to look at the course of disease and the incidence of adverse events like herpes virus and hepatitis virus, and learn the importance of B cells in these viral infections, and then you can do subsequent studies. So I think it's getting better, but the practical issues like biopsies, like additional samples, remain, because these are human beings. And consent and privacy considerations continue. But it's absolutely possible, and will be more possible when we have electronic medical records. Yes?
Hi, Karen Abudi from City of Hope. My question is on the phase 1 to phase 2 trials. When we're bringing stem cell therapies to patients, these are new trials, never been done before. There are no standards set. The FDA is kind of working with us to figure out dose. Nobody really knows. It seems like there's a lot of media attention and focus on these new phase 1 studies, the Geron study, the stem celling study, and there's a lot expected on the phase 1 study. Now, even though we keep saying the phase 1 study is purely a safety study, a feasibility safety study, it seems there's still a lot of pressure that you're going to have to show, maybe I'm wrong, that they want to see some indication of therapeutic efficacy to move to a phase 2. Sometimes what you're going to get from the phase 1 trial is pretty limited, but you have to start somewhere. You don't hear all these companies, I never hear the feedback. What happened? Where are these companies at? Are they in phase 2? Are they still analyzing the data from phase 1? I mean, it's years.
The one thing I would tell you, because I'm smiling as you're saying that, because I've said it myself many times, that phase 1 is only for safety. Listen, patients are waiting, so don't kid yourself that you can limit the questions that you ask in phase 1 to safety. The more excited the public is and investigators are about the potential for the therapy, the more people are going to ask those questions. The real question is, what do you do if you don't see it? Because the studies are so small, right. You're not looking at those endpoints. But I do think it is absolutely reasonable in phase 1 to ask about activity if your molecule should have a biologic effect. I mean, it is always important to pay attention. I mean, that sounds completely stupid, but the most important thing any clinical scientist does is absolutely look at every scrap of data you have. And in phase 1, you can get extremely important hints of activity. Some of the most important hints of activity I ever saw, now this is cancer, right? So that's a very specific trial, but we started seeing in the Rituxan studies tumor lysis syndrome, a very rapid breakdown of tumor with kidney shutdown. That was an adverse event. Well, I knew this was a powerful anti-cancer drug. You couldn't miss that in phase 1. With AntiVEGF, we had some bleeding in a tumor that had metastasized to the head. Again, this necrosis was happening at an incredible rate. You could see that in phase 1. So I think these hints of activity can't be overestimated. If you don't see anything, the question I ask is, what's the duration of therapy? Is it reasonable in a chronic therapy to expect short-term efficacy? Of course it's not. And the word efficacy shouldn't even be used around a phase 1 or 2 trial. But I also think it is not, it's not irrational for people to ask that when they're excited about the therapies. So it's inescapable. What I will say when you go into phase 2 is the dose ranging that you do in phase 2 is worth spending some time on. Because if you don't nail down 1 or 2 doses by phase 3, the complexity and the cost and the size of phase 3 becomes really difficult. And the only thing that I've learned in phase 2, and then if you do take 2 doses into phase 3, is make them as wide apart as possible. With Avastin, the 2 doses we end up using were too close together. And now, everybody wishes that we had them further apart, or more, but you always want more. But everybody's going to look for activity in phase 1. You can bet on that. Yes, there's another question, I think, same microphone.
My name is John Sladek. I'm a professor of pediatrics and neurology at Colorado, but more importantly on the grants working group of CIRM. And since you invited a tough question, Chancellor, I'll see if I can oblige. Early in your talk, you said something to the effect, discovery should reach the greatest number of patients. So that would tend to rule in Alzheimer's, diabetes, heart disease, et cetera, but it would rule out Fragile X and Down syndrome and the rare diseases like Batten's. And I don't want to misinterpret what you were saying.
Thank you for asking me that question. So that would be a misinterpretation. When I've talked about product development at UCSF, it's not uncommon that my colleagues think the goal is to get into the clinic, into clinical trials. And what I've seen at times with small companies working with academic colleagues is that you can stay in clinical trials for 2 decades. You can be in clinical trials for the rest of our lives. I think it is extremely important to have an approval, because no matter where that patient is, even if they're not at a big academic center in Colorado or San Francisco or Irvine, patient access is best with approved therapies. A clinical trial is an experiment. So I think the smallest patient group that can benefit from your drug is fine, but don't let it languish in the trials. That's my plea. And if your quest is to have an approved therapy, you have to ask yourself the hard questions, because you're not tinkering. And so I'm glad you asked for clarification. If it is a rare genetic disease, fine. But you still should try and get it approved by the FDA, because patients benefit more if you go through the rigor of proving it's safe and effective, and if somebody can sell it. Not because they make money, but because by virtue of commercializing it, they have to have production, distribution, supply chain, access. All those things are good for patients.
Thank you.
Thank you very much, Dr.
Thank you.
Desmond-Hellmann.